Please join me in welcoming to the Distinctive Voices podium, Dr. Carl Kottman.
Thank you, Jennifer. Is this on now? Yep. Okay, good. Yeah. This is one of these ones where it gets activated remotely, which is sort of a first for me at least. So anyway, welcome in this, sort of new darkness at this time of night. Kind of a little bit interesting to drive around in places you're familiar with and say, no, wait a minute, where the heck is this? So anyway, I'm pleased to be able to be here and share with you a message on cognitive health building and how we really can make a big difference in this country and even worldwide with promoting the value of exercise to individuals, particularly in the elderly. So today I'm going to share with you some recent data about how exercise can actually build brain health. Right now, Alzheimer's disease, which I'm sure everyone knows about, which is a sort of dementia, is increasing because of the aging population and some of the increase in risk factors. So this is going to essentially reach 30 to 40% of the population for those over 85, which is just downright scary. And we really need to do something about it. I'm not sure that there's going to be one magic solution, but there will be a series of solutions. And the sort of new guy on the block is lifestyles, which can have a profound difference. The startling fact is that there have been no new FDA-approved drugs for Alzheimer's disease since 2003, when Namenda, a cholinesterase inhibitor, came out, despite over 400 trials. That's not a very good track record, to say the least. And it really hit the pharmaceutical business as being really, kind of non-profitable area to be in. And some of them actually sort of went out of research because of the unlikelihood that they would succeed. Nonetheless, we are succeeding. And one of the big changes is to show that lifestyles have emerged as an effective strategy to delay the onset and improve symptoms of cognition as individuals age. And it's kind of a well-hidden story, but gaining traction. So what are some of the modifiable risk factors? Well, you can kind of imagine most of them. Certainly diabetes, which we hear about, and even the threat to the younger population, and maybe diabetes becoming even more prevalent because of lifestyle differences. Hypertension in the elderly is certainly an issue. Obesity, smoking, depression. Depression is actually able to affect cognition, of course. And then cognitive inactivity, and finally, physical inactivity. So what would you think is number one in that list? The last one? It may be the position is the way I probably gave it away, didn't I? Okay. So in the US, physical inactivity is in fact the number one. 40% of the US population is actually considered to be sedentary, which is really pretty amazing when you think about it, because it's defined as somebody that doesn't do more than a couple hours of activity a week. And that's really almost hard to do. But number two is surprisingly depression. Number three, smoking. And then finally, obesity, and particularly midlife obesity. Worldwide, cognitive inactivity from low education is higher and is number one in smoking too, because of the prevalence in many of the populations, including a lot of the European countries. And this is work that I really have to give a hand to Deborah Barnes and Christine Yaffe when they put this together. And it's not what you'd call bedside reading, but it's certainly a fact that is really quite startling. And most people write individual papers on each of these. They decided to take on the whole issue and came out with this exciting finding, which is something that we can pivot off of and build on. Okay, so physical inactivity is the number one modifiable risk factor. And I ran across this not too long ago, that sitting is the new smoking. So, you know, it's almost true, because smoking, of course, is a risk factor, and so is sedentary behavior. And I read these little newsletters that you can get from the Mayo Clinic, which I find really useful and accurate. And so this was the first place I saw this, and maybe they originated, and I'm not sure. Okay, so what I'm going to talk about this evening is essentially a three-part story. You know, it's kind of like chapters, and in this case, the book is only 3 chapters. But there's a lot in these chapters that's really worth knowing about and thinking about. So can exercise improve, whoops, gosh darn it, big fingers. Can exercise improve learning and memory? And how does exercise act on the brain? Interesting, because of course, you know, the brain is not out there jogging, your muscles are. So there's something by which the brain gets activated and can encode the effects of exercise, and we'll talk about that. And then, is brain aging caused by an epigenetic repression of gene transcription? And I'll talk about some of the epigenetic changes. Epigenetic means sort of past genetics of, you know, what you inherit is genetic, but what you can acquire in terms of reprogramming the DNA and the transcriptional machinery is really in the field of epigenetics. And that can be really long-lasting and very powerful. Then I'll talk about can physical activity drive gene expression in the human brain? And you'd say, well, how the heck are you going to do that? Stay tuned. I will tell you. And it's sort of it's an interesting, exciting story. And then I'm going to tell you about some new clinical studies on exercise that are underway. So, 3-part story, and we'll unfold them now. Okay, so exercise improves learning and memory. Initially, it was pioneered in animal models. And this is kind of the technique that was used. This is a radial arm maze where the animal is put in the center, and then there's a food reward in one of these arms. And the animal gets to find the food, and then there's a delay, and then the animal's put back into the middle and you know, has a number of trials to get and find the food reward. So it's sort of spatial memory. And it kind of reminds me of, if you don't park in the same place, but not even close to the same place, this is what you're doing. You're solving this little maze. And in UCI parking lots, sometimes that's for sure happening. So what we see here is that the exercised rodents do better on solving the task more readily and getting the target correct more percentage of the time. This type of effect has been demonstrated where running and exercise improves performance on several tasks. You know, there's been the Morris water maze, which is pretty much what I showed you, the radial arm maze, fear conditioning, object location memory, and pattern separation. And we can imagine pattern separation where you have positions of things that are identical, and you have to figure out the pattern and where is the novel, what's been changed, so what's novel. And again, that's sort of a practical matter that we see in everyday life. And these kinds of changes of exercise affecting a variety of tasks is really true in multiple species: mouse, rat, dog, and primate. And so it's really a principle. You know, when I first started working on it, I was working in mouse and rat. And then I was doing some studies in aged dogs in order to develop a diet that was good for the dogs and would maybe translate to people. And we tried then getting some dogs to run on a treadmill. And it was actually not that hard once you got them so that their feet didn't go slip in between the belt and the space in there. But once you trained them, they learned how to do it, and they were enthusiastic. They would wait, and when somebody would come to take them for their daily exercise, they would go out, jump on the treadmill, and start. And that's what we need to get more people to do, see? So, anyway, it's become a big principle. And my associate and I, Nicole Burchold, reviewed this several times, and even more recently. So, what about in people? Well, surprisingly, this didn't happen until more recently, and the first clinical trial was actually not until 2008. And it was really a very understudied area and came out in one of the best New England Journal of Medicine, which is one of the top journals in the medical field. And it consisted of 119 subjects with a subjective memory complaint. And they had been in 19 months of home-based physical intervention of usual care. So it wasn't really do nothing, of course, it was just do what you usually do. And they compared that to home-based physical intervention, being active and doing activities around the house. And it really wasn't that well controlled, and you would expect maybe it didn't work. But in fact, it did. It improved physical activity of like 110 minutes per week, significantly improved this Alzheimer's disease assessment scale cognitive measures 1.3-fold. Now, the reason that's really impressive, it's 1.3, is that is the same increase that was seen with the best drug available at the time for Alzheimer's disease. So the physical intervention was rivaling the pharmaceuticals at this point, and in fact still is doing as well as possible. So this was published, and then there was similar data came out by Laura Baker et al. In 2010, and she showed clearly that it wasn't just a global effect, but it was really one of the functions that benefited the most was executive function, that is decision-making and making choices strategically. So the studies, though, however, were relatively small, and the results, there was some variation. And we'll come back to this later on, because we're now doing a much larger intervention study for people with mild cognitive impairment, and I'll tell you and update you on progress on that. But Lautenschlager was the first in 2008. And okay, other studies have come in that are more descriptive in nature, and descriptive studies, if they're longitudinally followed, can be very persuasive. The Horder and group from Sweden followed physically fit women and showed that the physically fit women are 90% less to develop dementia. This was based on 191 women that were followed longitudinally for 44 years. So they had a really good baseline, and then they were looking independently at the amount of physical activity, and the ones that were high fit did so well that only 5% of those with high fitness developed dementia. And that's compared to, remember in that second slide, it was like 40% can develop dementia. So this is incredible. And in contrast, 25% of those with moderate fitness and 32% of those with low fitness in this study developed dementia. So this really got my attention in the whole field, because here it wasn't just making a little difference, it was basically only 5% developed dementia. That's basically close to a cure. So you think about it, the scourge of dementia, and maybe there's something simple like physical activity that can really bring this and turn it around and save the basically health tsunami that's going to come with so many people that could potentially get dementia at the current figures. Okay, so anyway, keep moving. That's the bottom line. And one of the things that's interesting is that these people are all happy, but it seems like there's also an attitude thing that develops. And I remember, when you go to the gym sometimes, I don't know, sometimes the people are happy, sometimes they're not, but I remember one of the first times was when I was in Copenhagen, and they had a fitness place you could go to. So being that I've been too much on airplanes and everything, I went and was there. And here these guys they had 4 treadmills all lined up next to each other. And they were just having a ball. They were joking around and jogging and, showing tricks they could do on the treadmill. And, and just, really, and they would even joke about it. Did you remember what I just said? Oh, okay, good. So, okay. So in other words, exercise can build brain health. It's becoming clear. Now let's go deeper into the story. And if this is going to be a real principle, there must be a mechanism. And oftentimes when you have a mechanism, it promotes expansion of research in the field, because then you can really get a hold of it, you can find alternatives, you can find strategies, etc., and you might be able to even take a lifestyle and pair it with a pharmaceutical to get more benefit out. So how does exercise act on the brain? Well, I was part of a MacArthur Foundation study group that was looking at the effects of how people could be promoted to age successfully. It was the Successful Aging Program. And one of the things that you came up with is that for those that did really well, there was physical activity, even including gardening and light physical activity, that seemed to correlate to the lack of cognitive decline. There are other things like self-esteem, a high value of self-esteem but that's a surrogate for lots of other things, and then social activity too. But so I got to thinking about it, and what the heck is going on, that how could physical activity basically translate to improving brain function? And so I went and did what you'd usually do. I looked in the literature, the scientific literature, and all I could find out is that if you look at animals or people that run, it basically increases metabolism. Well, you don't really need to read the literature to figure that out. So I said, well, so this was, sometimes I like to go in the backyard and walk around, and look out at the trees and everything and just think. And so I said, well, what the heck would really be the case that might give rise to some benefits? Well, at the time, one of the big molecules that was improving brain function and slowing down degeneration was this factor called BDNF. This is called brain-derived neurotrophic factor. It's actually a protein that keeps neurons healthier. It's kind of like brain fertilizer, if you will. You know, you sprinkle it on the neurons and they can resist damage. They can grow again after a little bit of damage, and they're just all of a sudden healthier. And, you know, so I figured, well, this is an interesting thing to look at. So, you know, I said, well, it'd be nice to look at the effect of exercise in some animal models to see what happens to brain-derived neurotrophic factor. So I went to the lab and, you know, approached one of my best postdocs and told him about this and said, would you like to work on, you know, the project and maybe get some exciting data? And they said, well, Carl, we're really busy. And so maybe when we're done. So I went to another one, same story. So after a couple more, because I was running out of people, I went to my first year graduate student, who was a woman that had a degree in physical therapy. And, you know, so she heard about this and she says, oh boy, I'd love to work on that. So she did some experiments and lo and behold, found that this BDNF was induced in the brain by exercise. And what was surprising about it is I expected it would be mostly in the motor systems, you know, because those are going to be more active when you're exercising. It turns out the big surprise was it was in the hippocampus, which controls cognition. And I said, oh, wow. That is turning, now the head has to turn around and you got to really think more about this because I was totally wrong. And the field was really wrong. So we published the paper, it got into Nature magazine, which is the sort of Time magazine of the scientific literature. And I mean that as a positive way too. So we did the experiments and lo and behold, it turned out BDNF was induced with exercise. Well, now all the postdocs want to work on it too, of course. So I said, sure, you know, we'll develop some projects. And when I first presented this at a scientific conference, everybody was a little skeptical. And then, you know, good science makes progress when people replicate your findings. And this was controversial enough that people went and tried it by themselves in their own labs, and it was replicated until now. There's over 1,000 replications of it. And it's one of the principles in the field that, you know, the brain can be reprogrammed and produce these health factors that are part of the brain's own healing. And it also helps learning. So, good news. And here's the story. Shawnee Nieper was the student that was a degree in physical therapy. And what she showed was that compared to sedentary animals, BDNF went up in the exercise animals, and it was kind of like a dose response, but eventually with high activity it goes down again. And this is probably what goes on with the marathon runners, because so you see them when they go across the finish line sometimes, and they're basically a very bad version of blue because of being anoxic. But in these rodents, they would induce BDNF, and we published this in several places and got replicated, et cetera. Okay, one of the things that is good is that you like to show that effects can be improved. We sort of wanted to look at what's called object location memory. And this is the car in the parking lot story. Here's the animals that get put into a big arena with a couple objects, and then 24 hours later, one of the objects gets moved, and the animal has to tell which one is moved. And the animals are curious about things, so they will naturally go to the new object in a new position because they're very sensitive to their spatial environment and can really do this spontaneously. So this doesn't require any particular training. In fact, you can do it in one trial and get reliable data. So it's a very nice task in that there's not a lot of unnatural, like swimming in a water tank or anything like that that's cold water, et cetera, which would be, I think, pretty unpleasant. So, and the advantage of these types of tasks is they could be made more difficult as well. So what we did then is to show that BDNF is basically increased with exercise. The 24-hour retention is improved, and with that exercise, BDNF messenger RNA goes up. And if we then use what's called an inactivating RNA or a silencing RNA you can inject the BDNF, but if you've got the silencing RNA around, the BDNF doesn't really work and get a hold of the brain system that's needed to be activated. So you can then show that BDNF is really the critical molecule underlying the exercise benefits by using the siRNA and showed that really doesn't, it takes away the retention and it blocks the BDNF effect. So that was one of the first stories that really showed that yes, indeed, there's one molecule that is the readout in the final common pathway for the benefit of exercise in the brain. Pretty big story, actually. And this was done by a graduate student that worked with me in 2013 and has continued in a career roughly in this area from there. Okay. So one of the interesting things is, if you're a scientist and a lecturer, you sometimes end up more on the airplane than anywhere else. And I was getting tired of this sitting around in airplanes and figured, and maybe I'm losing the benefits of my exercise program, while I'm sitting around. And so I kind of got to think, how long is exercise really effective? You're going to do this, but maybe you need to, if you skip a few days, maybe it's not going to, the effects are going to go away. And, muscle conditioning, for example, doesn't necessarily last forever. So how long does the cognitive part of exercise? So what we did is investigate the possibility that there's a molecular memory for exercise. There's a molecular memory for learning lists and items, but maybe there's similar vocabulary and mechanism to build exercise. And so we began looking at that, and so asked the question of what is the role for frequency, duration, and intensity of exercise. And I won't go through all of the data on that, but I'll go through some of the key findings. Is if you have an animal that has exercised for 2 weeks, by 1 week afterwards, the levels go back down again. So they're not that long lasting. And, by that, you kind of have trouble thinking about how's the benefits so profound. Well, it turns out that if you just give a small amount of exercise, like two days here, which doesn't by itself induce any real BDNF increase, but that if, in fact, there's 1 week of rest, it goes down, and then if you give a brief hit again, or a reminder of the exercise event of essentially 2 days, so exercise for 2 weeks, take a rest for 1 week, and then give a 2-day reminder. It goes back up again. That's pretty cool. So in other words, when you miss a day or two, you've got a window, and then you can get back and get all the benefits back without having to go through the whole conditioning thing again. And this has helped, formulate some exercise programs in the field now, and seems to be holding up really very nicely. So BDNF has a molecular memory for exercise, and it translates to the cognitive level. So what we found is that if you look at 3 days, it's still present. It's present at 7 days, but by 2 weeks, it decays. Well, that's not bad, 2 weeks. That gives you a lot of time to reschedule yourself and get back into it. If for some reason you can't have exercise, because of schedules and other problems. So you got a little window of opportunity to bring all the benefits back again for cognitive performance particularly. Okay. So this may explain then why exercise can be effective in humans despite widely varying exercise practices and inconsistencies, skip days and even weeks. And this helps explain why in some surveys, like initially where I started thinking about this, is the Nurses' Health Study looked at the effects of exercise on cognitive health of nurses. And they looked across all the places in the United States, and the data was gathered. And then I looked and broke the data down because it reported it by regions. And I said, well, in Montana in the winter, there's no way in the world that people are getting the same activity patterns as they would in the summer. Or at least that's the assumption, and that turns out to be the case. But what they're doing is that there is a period of grace, if you will, and the body and the brain have a molecular memory to benefit the brain's health. It's really pretty cool principle when the brain is looking after itself by working a little bit. Okay. So now let me go into some of the epigenetic mechanisms and how it affects aging. Okay. Cognitive decline precedes neural degeneration. You think that degeneration is what's causing cognitive decline, but it turns out that that's not quite the case. And that if, in fact, you look at some of the studies in transgenic animal models of Alzheimer's disease, that the cognitive decline comes before any detectable effect on degeneration. So there must be another mechanism. And I know I ran this by a couple of my colleagues, and they said, you might be right, but what? And so I thought maybe there's a repression of gene transcription that develops with age and impairs neuronal function. If so, then this repression might be reversed and might account for some of the age-related cognitive decline that is recoverable. And that would be really good news, because then it wouldn't be degeneration, it would be just repression. We then looked into this, and epigenetic controls the accessibility of the genes for transcription. You just take my word for that, because the control of the readout of the gene is controlled by sort of a thing called epigenetics, or that's past genetics. And one of the important repressive factors is a mark that's histone 3, lysine 9. When it gets trimethylated, it becomes repressive. And there's a colleague of mine that had developed a drug for inhibiting this particular formation. And this is where, sometimes serendipity kicks in. As I was at a dinner, and it was one of these university dinners that we all just adore. And I was sitting across the table from him, and I said, well, Larry, what are you up to these days? And he started telling me about these compounds that inhibit, the epigenetics on the histones that control transcription, or the reading of it. And so they said, well, boy, this is kind of interesting. So I went back and thought about it for a day, said, I really got to follow this up. And so I convinced one of the postdocs, who initially was a little bit skeptical, but she said, okay, just so he stops bothering me, I'll go over and do it with him. And so we got some of the compound, and she said, well, came back a couple days later, she says, it worked. It actually recovered memory in these old animals that I injected, and it was really quite remarkable. And so with one injection, they went from cognitively impaired to normal. Wow. Yeah, no joke. I mean, that's pretty exciting. So there is this novel compound that reduces the histone methylation and is undergoing current additional developed by a really good chemist at UCI called by the name of Larry Overman. And this inhibits a known target, which is the histone methyltransferase that drives the formation of this particular inhibitory mark. So right now, there's a prototype in phase 1 trials, so that's sort of good news, and we'll wait and see how it goes along. It also turns out to be useful for promoting healing in the skin level, and so it has a number of benefits that may encourage its development. Okay, so with age, then we began to look at this factor, and with age in the hippocampus, the compound increases. It does not, though, in every brain area, like the cerebellum, which is involved in just motor function. But in the hippocampus, which is involved in cognition, it increases, and it may be one of the factors that is inhibiting learning. And so what we did then is to try and inject the compound that inhibits the histone methylation, and found that relative to a control, that the injected animals can do this little object recognition task really fine. These are about as good as it gets. So, but if they don't have the compound, these older animals can't really do the task. These are 18 to 20-month-old mice, and they're really, they only live for another half a year. So these are really old mice. But the fact that they can be restored is exciting news indeed. Okay, so it restores cognitive performance. Also in a mouse model of Alzheimer's disease, which is this 5X, Alzheimer's disease mouse, and this is the way the animal tests out. They cannot do the task at all. But if we give them this compound, they can do the task. So it's really rejuvenating the aged brain to make it perform as if it's younger again. Cool finding. So we can imagine that when this becomes available, it could very well help rejuvenate people's life and lifestyle. So, okay. Now, what about the gene level? I want to go and talk about, will physical activity regulate gene expression in the human brain? And so this was sort of a fantasy of mine that maybe we can figure out, because I've been doing studies on gene expression in rodent models, but I always wanted to be sure and find out, what about the human? And so, I knew that one of my colleagues at Rush Medical Center in Chicago had been doing some studies on longitudinally following people and measuring their cognitive performance. And so I thought, well, maybe he collected some postmortem tissue. He's a very thorough investigator and never let anything go to waste. So indeed, he said, I've got postmortem tissue. And I said, well, do you have some health history on them? And Do you know by any chance how much activity pattern they have? You know, like, do you have a record on their participating in a Y or a health exercise program for elderly, et cetera? He says, I can do better than that. He had cases that he followed longitudinally and had postmortem and brain tissue available. So we got a hold of some of the tissue from these people that had actigraphy. Now, this is a pedometer that people were wearing. So he knew precisely what kind of activity patterns they had. And this is the way it looked. They had the low activity and the high activity. So what we did then is compare the gene expression patterns of these individuals versus these. And so we could find out how activity reprogrammed the brain. And, you know, I have to admit, when I did this, I wasn't convinced that there would be anything in this at all. You know, it might be one of these nice ideas that doesn't work. But in fact, it turned out that when we ran some microarrays, which scans the genome and gives us every gene that changes, that the individuals that had high activity compared to those that are aged and an AD set were really comparable. And we found that, in fact, the majority of genes that had physical activity, instead of going down with aging, they went up, or it didn't change. So basically, what was so surprising is that physical activity was keeping the gene expression patterns as if they were younger, you know, which is pretty amazing when you think about it. And so the majority that showed decreased expression showed increased expression with physical activity. So thus the physical activity was rejuvenating the gene profile of these individuals. So what's the function of these genes that changed? You know, is it exotic? Because there's a lot of genes that we hardly know anything about. You know, we think we know a lot. We do know a lot, but we don't understand all of them. So are there specific functions? Well, two of the key things that change with age are synapses, and those are lost and compromised with age, and particularly Alzheimer's disease, but with age, and then mitochondrial energy production go down. So what happens then to the functions of the genes that decrease with increase with physical activity? Do these categories get affected? Because they're known to be compromised with aging. And what we found is that, like in the mitochondria, for example, the genes in the energy production machinery, all these blue genes are increased, and you can see that the key functional areas in the electron transport chains that make the ATP and gives the cell energy are going up. You know, really quite remarkable, and just not in one little complex, but in the key ones, the initial entry complex and the one that makes the last of the ATP. So these are, we're down with aging and up with physical activity in the energy production machinery of the brain. And in fact, when we looked at the effects of physical activity with aging and AD, from young to aged to AD, the expression patterns are going down, and with physical activity, from the low activity to the high activity, the patterns are going up. So it's basically counteracting the downward decline that one sees with aging and Alzheimer's disease. And these were on some of the key synaptic genes that are listed down here. So this has become quite a powerful story. So the next stage is really, we need clinical trials to really document not just the small studies and the preliminary studies, but we need a serious clinical study to follow the effect of physical activity on elderly people with a little bit of cognitive decline. So if we can take a little bit, maybe we can slow it down or stop it, and then document this. So in fact, there is a study now called Exert, which I'm part of, together with Laura Baker, who's from Wake Forest University. And we partnered up and are doing this study, which Exert stands for the exercise trial. It's not too complicated. This was her idea, and I thought that was really clever to be able to come up with this. And so what it is we do need a cost-effective strategy to translate exercise intervention to the community setting, because it's nice to publish it in scientific journals, but let's do something with it. And so UCI and 14 other universities around the country are funded by the National Institute on Aging to partner with the YMCAs to essentially carry out the exercise study. So, you know, that's a perfect outlet to translate it to the community, because that's what these people do. They're in the community. And once they finish the program at the Y, you know, with the exercise program, they've got a natural community environment to continue in however they wish. And then they've got the incentive that we hope will make a big difference in the exercise patterns of the elderly population. So these people complete their exercises at the Y, and under the supervision of a studied certified trainer in the first 12 months, and then independently in the final 6 months. And so we're following their cognitive performance, doing brain imaging, and doing biomarkers through this. And the study is still ongoing. It's a 300-person study. We've enrolled now 240 some individuals, and we had a goal to hit the 300 by December, and we're going to make it now. It was really hard initially to get subjects into this because it was 4 days a week of a commitment, and you know, of a couple hours a day. So it's not a minor thing, but in fact, once the people get in, there's almost no dropout. So they like it. They just are hesitant to get into it, but once they're in there, they enjoy it. We'll find out how it goes. So this is the study. It's a sort of a year-long study with a 6-month follow-up, and we do a walk test to get their physical fitness when they enter, do brain imaging, cerebral spinal fluid collection, and then cognitive assessment at 0, 6 months, and 12 months, and 18 months to see how it goes. And then we'll try and follow these individuals even longer than that, but we'll have to get another grant for that. So always another challenge. We had some spontaneous comments from some of the people participating, and I wanted to go through one of them. Testimony from an individual that was in the Exert trial. Participating in the Exert program for just about 2 months, I find to be an astonishingly transformative experience for which I am profoundly grateful. With Exert, the transformations have been bodily and energetic. So who am I now? Tall, vibrant, upright, energetic, walking the treadmill twice a week, 45 minutes each time, walking out of doors as yet less often for shorter periods. Huge gratitude to and for everyone involved in Exert. Incredible. I mean, if you had a speech writer, it couldn't have been done any better. So this is the way the woman felt about improving her physical condition and doing this particular activity. And, you know, I was very excited and, you know, really actually even surprised myself that how positive this was. So, so let me summarize then. Exercise increases BDNF in the hippocampus, the sort of brain fertilizer. Exercise builds a molecular memory, which allows rapid memory reactivation to essentially preserve the cognitive benefits of exercise. In aging humans, physical activity is associated with increased expression of genes regulating synaptic and mitochondrial function. And these are the targets that go down with age, and they get rejuvenated by exercise. In aging, the repressive mark that I told you about, the histone 3 lysine 9 methyl trimethylation increases and impairs cognitive function. I showed you that there's a drug that knocks this down again, that restores learning, a UCI collaborator that I had mentioned, and that reducing H3K9 trimethylation restores learning in aged mice, and we're on our way to try and translate this to people. Thus, H3K9 dependent gene expression may be one of the key factors that drives brain aging, and it's not then degeneration, it's repression of genes. And that, if we can then find the key factor, unlock the door, we will be able then to keep more people functioning at a higher level before they have to, just rebuild the brain circuits. There's really going to be a big, big challenge. So with that, I've had a lot of people involved in the study. My coworker, Nicole Birchold, who's been a really cornerstone of this. Chris Butler, who did a lot of the human work. Shika, who did some of the early animal studies that really put this on the map. And then a whole series, Laura Baker, who's running the clinical trial, Exert, with me. David Bennett, who's provided some of the tissues. And then finally, some common facilities at UCI and elsewhere, and a bunch of collaborators. So, thank you so much.